All right, the third section for chapter 7 now, we're looking at the synapse. So the synapse is a functional connection between neurons and the cell that it signals. So within the central nervous system, this occurs between two neurons or several neurons between each other. In the peripheral nervous system, this occurs between a neuron and some type of muscle or gland. So within the muscle, what we have is a special term called the neuromuscular junction, which is a synapse between the two. Synapses can be either electrical or chemical, and we'll go through those in a moment. And we'll have the special terminology where the first neuron is called a presynaptic neuron. And then the second neuron is going to be the postsynaptic neuron. So with the electrical synapses, these are a lot less in common than the chemical synapses. And so we find this within smooth muscle and cardiac muscle, and then also some regions of the brain and between glial cells. And the important thing for this is that these help to contract as a unit. So for instance, for smooth muscle, we see this with peristalsis and digestion, where we have movement and contraction of that muscle that occurs at once. And then within the cardiac muscle, for the heart to beat as a synctum or as a unit. So with electrical synapses, these are due to gap junctions that occur between cells. And so when they have closing or opening of these channels, then that's what will cause them to contract as a unit or not. Chemical synapses are more prevalent, and this is due to the release of neurotransmitters from an axon terminal. And so we'll go through some of the anatomy for terms, and then we'll see a picture in the next slide. All right, the presynaptic neuron is what conducts action potentials toward that synapse. The synaptic knob is what contains synaptic vesicles, typically of neurotransmitters. Synaptic vesicles then are what hold neurotransmitters, and these are going to be chemical signals that then are going to cross the membrane via exocytosis. And then the postsynaptic neuron, these are going to be that have action potentials away from the synapse. And then the synaptic cleft is the space between the pre and the postsynaptic neurons. So let's look at this. So here in this example, this is going to be our presynaptic neuron, okay? And then our postsynaptic neuron, or cell, oh, there we go, is this cell. Okay, so this would be like number 1, and this would be number 2 cells. All right, so what happens when we have a action potential invading the terminal is that the action potential is going to propagate, remember, down from the axon and then come into the nerve terminal, axon terminal. And so that is going to then open voltage-gated calcium channels. And so calcium, again, by net diffusion, is going to move from outside of the cell into the cell. And so calcium then is going to bind to certain proteins and also increase the volume of that synaptic bouton, where we are going to have these synaptic vesicles that are going to start to migrate toward the membrane. And so we have different proteins too called snares that are responsible for docking these vesicles so that they're ready to go once an action potential comes out. And so that causes fusion with the plasma membrane of that neuron. And then we'll have exocytosis of that neurotransmitter. And that's going to migrate within that synaptic cleft and then bind to receptors within that postsynaptic cell. So if we look at electron microgram here, what we see is that here is that terminal bouton of that axon, and you can imagine this process needs a lot of energy. So we find a lot of mitochondria also within those axon terminals. So here we have these little circles are those synaptic vesicles, and then that very small space that we have here is the synaptic cleft. So this would be our presynaptic, and then here would be our postsynaptic cell. And in this case, this is the neuromuscular junction, so this would be with skeletal muscle. So there are two types of synapses that we have. Either they're going to be excitatory or inhibitory. And so this means that it's either going to excite that postsynaptic neuron or cell or inhibit that postsynaptic neuron or cell. And so this is based on what neurotransmitter, NT, is going to bind at the postsynaptic cell. Okay, so it has to depend on what is the neurotransmitter and what channels that will open, because as we learned earlier, we have both excitatory and inhibitory channels. So if we begin with our excitatory synapse, so this is caused by opening sodium and calcium channels that causes depolarization. And so on that postsynaptic cell, that causes an excitatory postsynaptic potential, or EPSP. All right, and so here, when we look at this figure, what we have is, here our neuron is at threshold, and then that postsynaptic cell has a few of sodium or calcium channels that have opened it. However, since we haven't hit threshold, that means that an action potential has not been generated, okay? But we see that there's a slight depolarization when we have those ions binding within those channels and those channels opening. However, that was not large enough to elicit an action potential. Though, if we had enough ions, that could cause an action potential to be generated. Now, an inhibitory synapse, we have opening of potassium and chloride channels, and this causes a hyperpolarization, so it becomes more negative, and so this is termed an inhibitory postsynaptic potential, or IPSP. Where now if we look at this figure, so again, voltage is on the y-axis, and our resting membrane potential for that postsynaptic cell is at -70. But now what's happened is that activation of these channels causes a more hyperpolarized state, so getting farther away from threshold, and so it's a lot harder to generate an action potential then. Now, with our neurons, it's not just a one-to-one relationship where one neuron is acting on another, or one presynaptic on one postsynaptic. Rather, there can be, on one neuron, there can be thousands, maybe tens of thousands of other neurons that are acting on it, okay? And so for that postsynaptic cell then, what we can get into are temporal summation and then spatial summation. So with temporal summation, this is where we have lots or successive waves of neurotransmitter that are released and added together on an initial segment of the axon. Whereas spatial summation is due to the convergence of signals onto one postsynaptic cell. So that means all of the excitatory and inhibitory postsynaptic potentials are going to be added together at that axon hillox. All right, so here let's look at the influence on the postsynaptic cell by three different types of neurons, of presynaptic neurons. The first one is excitatory, the second one is excitatory, but the third presynaptic neuron is inhibitory. So if we look at panel A here first, what we have is, on the y-axis, we have the membrane potential for that postsynaptic cell, okay? And if you recall, membrane potential stays at negative 70. Okay, so this first panel here is showing that with excitatory presynaptic neuron number 1, its neurotransmitter generates a depolarizing effect for that postsynaptic cell. However, the frequency at which it fires is slow, and so it does not generate an action potential for that postsynaptic cell. Now, with temporal summation here in panel B, we have that same neuron firing, and the only difference now is the frequency at which it fires, and thus releases its neurotransmitter. And so if it is quick enough, what can happen is that those responses can build on each other and can cause a great effect, leading to an action potential for that postsynaptic neuron. Panel C here, what we have is now excitatory presynaptic neuron number 1 and presynaptic neuron number 2, where if presynaptic neuron number 1 fires and then releases its neurotransmitter, followed by excitatory neuron number 2 a little bit later releases its excitatory neurotransmitter, that alone will not generate a response. But if they work together at the same time and release their excitatory neurotransmitter, then that's going to cause depolarization that will be able to meet threshold and thus generate an action potential for that postsynaptic cell. Now this last panel of here in D, what we have is now excitatory presynaptic neuron number 1 and inhibitory presynaptic neuron number 1. So you can imagine with the excitatory neurotransmitter release, that's going to cause slight depolarization. However, now the inhibitory presynaptic neuron is releasing an inhibitory neurotransmitter, and so those two are going to cancel each other out and will remain at or near resting membrane potential. So it has a harder time then to reach threshold for that action potential to occur within that postsynaptic cell. Another way that we can have an effect on the postsynaptic cells is by presynaptic inhibition or facilitation. So this is where a third neuron will influence the activity between a presynaptic and a postsynaptic cell. So in this example, what we have is C, this is the postsynaptic cell. D is an excitatory presynaptic cell, and A is also a presynaptic excitatory neuron. However, this figure of B here is an inhibitory neuron that's acting on this presynaptic neuron of A. And so that can influence then if B is firing a inhibitory neurotransmitter, that will block the effects of the excitatory A neurotransmitter, so essentially canceling each other out. Then, if that happens, in order for an action potential to occur for neuron C here, it either has to be by the increased frequency of neurotransmitter release in D, or A has to overcome the inhibitory response from B. And so this is very common, and again, like I said, we have like 10,000 neurons acting on just one postsynaptic neuron. And so we see this physiologically with the action of endogenous opioids to reduce pain, and it inhibits the release of substance P, which is the main neurotransmitter that promotes pain transmission.